Okay, so my name's Meena Kumari, and I am a co-investigator of the Understanding Society dataset, and I look after the health and biomarker data in Understanding Society. So today we're going to focus on the genetic and protein data that are available in the dataset, but I'll give you a background. I'll start off and just give you, I don't know if everybody here knows the Understanding Society dataset. So the first half of I'll be talking about half an hour, and the first half of my talk, I'll be talking about the data that are available in Understanding Society, and then I'll talk a little bit about the biological data that are available. So this is an overview. So the health and biological content, and then I'll just do a brief discussion about terms, because there's lots of jargon and lots of terms used with the biological data, and it's sort of changing over time. And so I'll just give you a definition so that if you come to these data types or these data sets or this literature, you can have an idea of what people are talking about. And then just examples of how the data have been used in ways that are interesting for social scientists. So Understanding Society is the UK Household Longitudinal Study. It's a survey of individuals, and it's representative of the UK population. And we interview everybody in the household. So it's a household panel study. We randomly select households across the UK, and we collect information from everybody in the household. So that's adults and the children, and there's a core sample of participants, and we interview everybody annually. It's a basic design. It's similar to a number of international household panel surveys, so PSID, SOAP, HELDA. So there's you can do lots of comparative analyses. I think the strength of Understanding Society is that we do have the biomarker data that I'm going to talk to you about, that a lot of other studies don't. So that kind of makes us unique and special. So the study started in 2009, and it builds on the British Household Panel Survey. And there's lots of different types of information that are collected. So what people are experiencing, attitudes and identities, well-being. And obviously with the interviewing participants annually, we're getting an idea of how these things respond to policy changes, key events in people's lives. A big event has obviously just happened to all of us in terms of pandemic. And so we can see what was happening to people before the pandemic. We'll be able to interview people afterwards. We'll be able to see what's happened to them afterwards. So there's lots and lots of research possibilities, measurement of change over time. And because we're interviewing everybody, you can examine what's happening to people across the life course. So it's a really important survey to think about how policy environments are changing, what's happening to people, what's happening to people in the face of changing kind of norms and environments. So that's sort of kind of one of the strengths of the survey. It's composed of four kind of main there's four main parts of the survey. So the general population sample, which is a sampling of the general population. Sorry, I just had to repeat myself. So it's 30,000 households, and we've got families across the UK, as I said. There's an ethnic minority boost. There's actually two minority boosts in the survey. So we've got an ethnic minority boost, which is sort of established ethnic groups, really. Indian, Pakistani, Bangladeshi, Caribbean, African ethnic minorities. And then recently we've had a migrant boost, which was the Eastern European migrants. So there's a sort of the newer groups that came, and the more established groups are boosted in the survey. And the survey incorporated the earlier British Household Panel study, and we have biological data from the general population sample and also the British Household Panel survey. There's a test bed of participants that we do methodological development and research in, called the Innovation Panel. And we also have some biological information in that panel now, because at IP12, I did an experiment looking at how to collect biomarker data from different, from using different modes, so nurse, interview, and web-based. So there's biological data in 3 of these subgroups of understanding society. So this is a long schema of the data set. So as you can see, the red rectangles are where we collected biomarker data. So we collected it in wave 2 from the UK household, from the general population sample of understanding society. So we had 1 wave from that set of participants before we collect the biomarker data. And we collected biomarker data from the BHPS sample 3 of understanding society. So we had 20 waves of data before we did that biomarker collection. So there's lots of possibilities here in terms of doing analysis with looking at biomarkers with the data set. As I said, we measure the richness of people's lives as core data that we collect from participants every year. So education, employment, health and well-being, civics, income, lots of sort of core things that we're collecting all the time. And then a rotating set of topics. And we do that because it's impossible to collect everything at every wave, and we have to think about how much burden we're putting on the participants in terms of their data collection. So we interview them for about an hour and collect all of these different bits of information. The health and well-being, or the health element of the study are collected in 2 different ways, really. One from questionnaire, so asking people about their health. At wave 2 and 3, we have information on things like prescriptions. At every wave, we ask a whole bunch of chronic conditions. We ask at every wave, we're asking or administering a questionnaire on mental health called the General Health Questionnaire. At every wave, we ask about mental health and physical health functioning. And then, as I said, at waves 2 and 3, we administered a nurse visit. So where we collected a number of different objective biomarker data, so heights and weights. A nurse went to the participant's home and did a set of biological measures from the participants. So this is just more detail about the sorts of things that we've collected from the participants in terms of their health, the conditions, the SF12, the GHQ12, health behaviours. And we have tried to consented the participants to data linkage, although we've not been tremendously successful in actually doing that linkage. So we're trying to do that at the moment. We have data linked to the Scottish survey, to the Scottish health data, and we should be having data linked to the Welsh data as well. We just don't have English linkage at the moment, and we're trying to do that. And this is the non-health content, so I won't read it out. There's obviously lots here, and we're recording this, so you can come and have a look at all the different things that we've measured in the data set. And as I mentioned last year, the year before last year, the pandemic came upon us, and we were quite nimble in understanding society in that we managed to administer a set of questions. I think we were the first longitudinal study, national longitudinal study, to administer and deposit a data set on about the pandemic. So lots of information about symptoms, and then all of the things that we thought might be have resonance or related to what was happening to us last April in terms of lockdown. So there's lots of things here that we've measured, and we've measured again and again. So we were initially measuring monthly, so April, May, June and July, and then every other month, so September, November, January this year, and then March this year. And we've just we administered another survey in July, and we've just administered another one, which will be our last one, as a lot of the content has migrated into the main data collection, so that we don't need to do these extra COVID collections. So in terms of the long-term content, the questions are, we repeat lots of questions, we rotate modules, we are experimenting with using something called event-triggered data collection. So if something happens to a participant, an example of this is the pandemic. So the pandemic happened and we were nimble, and that we could ask lots of sort of focused questions about that. We're thinking about doing that for other things, you know, if you get married, if you have a baby, if you retire. So thinking through whether we can do these sort of event-triggered collections and age-triggered collections that are focused on the particular event or that's happening to you. So this is the long-term content, and you can see how detailed it is, and also, but also how it can mean that not everything is asked at every wave, and that you kind of need to know the data set and go and have a look and see what's asked when, and so how you can think about how to analyze the information that we have in the data set. So this is what went into the nurse assessment. We collected heights and weights, respiratory function, blood pressure, and then we collected a blood sample, and it's the data from the blood sample really that we're going to focus on in the next hour or so, in that we measured a number of analytes, and we collected DNA, and we're sort of interested in how we use that information and whether that information can be used in a social science context. So we were quite careful about what we measured. We sort of thought about what would be most useful. So our criteria were around environmental factors and whether they might be associated with the biomarkers that we wanted to measure, whether they were going to help us understand the pathways by which the social and health might be linked, whether the health outcomes that we were trying to capture affect a reasonable proportion of the general population, and you know, there's a reasonable prevalence in terms of the health outcomes. And also we're kind of limited by the because a nurse goes into the participant's home and the blood sample is sent through the post. So it means that not everything can be measured because some things aren't robust to that sort of collection. And so we were looking at core markers for main disease and things that we thought that would be useful either on their own or in combination to help us understand sort of how the environment gets under the skin. So those are the sorts of pathways we were thinking about. And this is a list of those, the analytes that are already available in the data set. So you can go to the archive now and you can have access to these data. They're already there. They were measured from the sample, and I think they were deposited a few years ago now. So, and there's lots and lots of examples of people using these data and doing lots of interesting analyses with them. So, you know, why would a social scientist be interested in biological data? As I mentioned, we focused on trying to understand the pathways by which social circumstances and health are linked. So sort of really understanding, you know, what is it about the environment that might be impacting our health, sort of which things, sort of pollution might be working down one pathway, occupation might be doing something else. So sort of thinking through all of those different processes. We're also thinking about subclinical markers of disease and objective markers of disease. So I can give you an example of how we did that now, but also thinking about the sort of the clinical iceberg. People will often walk around sort of on the pathway to disease and not know it. A few years ago, you would, half of people who had diabetes didn't know it. That's not true now, because it became a focus of clinical care, as it were. And so there's fewer people who don't know that they've got diabetes. But if you don't know it, you're walking around with it for a little while, and it's something that's coming down the road. So that's quite important in terms of understanding population level health and identifying populations at risk. And the other thing that we're particularly interested in or thinking about was that a lot of social science is different to other sort of disciplines in that it's open science, and again that's changed over time, but it wasn't, and it still actually isn't quite normative to be depositing data or making it available. And having lots of biomarkers in a social survey means that it's an example of a more sort of open science, in the sense that anyone can go and get these data. As I mentioned, the data that I've talked about so far are available in the archive, and you can go off and do your own analysis. It's not dependent on me. And you know, I'm sure people have lots of interesting ideas that and things that I would never think of, and so that's a real strength of the data set and this approach. So today we're talking about omics. So we've talked about genomics, proteomics, epigenomic. So there's lots of omics that have become it's sort of a term that people have started to use, and it's a term that's used for a slightly different approach to the way that we do these things than has been done in the past. So it's a totality approach. So you hear about people doing GWASs, and GWASs are genome-wide association studies. And in a genome-wide association study, what you've done is you've looked across the whole genome and looked at it, and you're interested in looking across the whole genome and your phenotype or your outcome of interest. And so there's a whole branch of science now that's moved to kind of down that direction. So you have genome-wide scans, you have epigenome-wide scans, and these data are available in Understanding Society. And what we have done, and there are going to be talks after me that go into much more detail than I'm going to do just now, but this is a sort of introduction, is that we have measured signals across the entire genome. So these things called single nucleotide polymorphisms, we'll explain that later. But we have data that has measured 14 million of them across the genome, and you can look to see how that's associated with your phenotype of interest or your variable of interest. We have measured methylation, which is a measure of something called epigenomics. And again, we've measured 850,000 sites across the genome. So you can look at that with your outcome of interest. And we've also are going to make available 100 proteomic data. So we've measured 184 proteins. That's actually a very small subsample of the potential number of proteins that are available in biology, which is thousands and thousands. So 100,000 proteins are probably measurable or available or in biology. But we've measured 184, and you can look at those. We'll make those available soon, and you can look at those and your phenotype of interest. And so people put these things together and talk about multi-omics or integrated omics in terms of these types of data. There are other terms that you might come across in this new omic world, and so people do something called exposome approaches, and that's really everything that's not genetics or genomics in terms of your exposure. So everything you've ever been exposed to is the exposome, and when you go to that literature, what what at the moment it appears like it's everything that you've ever been exposed to in terms of chemicals, really. So the pollution literature often talks about it being the exposome, and it's really all the different chemicals that you might have been exposed to. I think in the long run, if we really are serious about everything that you've ever been exposed to that's not genomics, a data set like Understanding Society becomes really important, because we've measured lots of things really well, and we've got everything that, you know, we've got an idea of kind of life course exposure to things. And then I came across recently this new term, this culturomics, which I don't actually know very much about, but it's the use of big data to understand human behavior and culture. And again, it's sort of trying to capture the entirety of an exposure. Having said it's a new term, it's actually used in a different setting as well. If you're a microbiologist or a basically lab scientist, culturomics is often the things, the sort of microbes that you have ever been exposed to. So sometimes new terms come in to use, and they are or are not associated, you know, take on new meanings. So it's all really good fun, actually, sort of these new worlds. So I'm going to quickly just do a one-pager on genetics. We're going to have a much more detailed and better explanation of genetics in the next presentation, but just as an introduction, so that you've got, there are lots of terms, as I said. So we've measured SNPs in Understanding Society, and methylation in Understanding Society. And so what that is, so as you, in my one pager of genetics, so you know that DNA, I hope you do, is found in your cells. It's in the nucleus of your cells, and it's sort of bundled together in chromosomes in the nucleus. So you've got about 6 foot of DNA in every cell in your body, and it's all wound up very tightly in these chromosomes, and it's made up of base pairs. So these chemicals called cytosine, guanine, adenine, and thymine. And together, that's sort of your alphabet, as it were. And you read that, and they're sort of packaged up in genes, and those genes make proteins. And that's really what we're going to be talking about today. So and methylation, which is the sort of a way of dialing up or dialing down how much of the genes are expressed to make the proteins, are chemicals that come in and sit between these C's and G's, so these cytosine and guanine, to impact how much they can, the machinery of the cell can come along and influence how the gene expression and the protein that you make. So I would say across this diagram, we have measured the C's and T's, the C's, G's, and A's, which are the SNPs. So those might be the same or different between people and impact what protein is being made. We've measured methylation, which impacts how much of the protein is being made, and we've measured 184 proteins. So that's sort of what we're going to be talking about today. As I said, the way that we use biomarker data is to sort of think about the pathways by which the environment and health are associated. And in the past, we've gone down this sort of candidate approach, and that sort of contrasts with this omic approach. So we have written lots of papers using this sort of candidate approach. So if you're interested in, if you think inflammatory markers, for example, are important for the mediating the association between the social environment and health, you know, we have already 2 proteins that indexed in inflammation, and we've written about how they're socially patterned in the past. So this is a paper that was written by Apostolos Davilas and Michaela Benzal and myself in 2017, describing how in the different age groups you can see a difference by educational attainment or income in people who have lots higher education or no qualifications, and that the difference in inflammatory markers between those groups emerges in our participants who are in their 30s and remains there until they are in their 80s. And so this diagram sort of gives us a description of two inflammatory markers. Other people at the same time were trying to, maybe we haven't measured the right inflammatory marker and have looked at a wider set of inflammatory markers. This paper by Castanier measured 28 inflammatory markers and looked at occupational differences in those inflammatory markers and created a score and to help us understand which inflammatory markers might be most pertinent to social differences in health. And so there's a sort of, you can see a sort of move from picking out one or two things that might be interesting to looking at things in more detail, to trying to look at everything and seeing what comes out. And here again, these two papers decided that inflammation might be quite interesting and looked at methylation of the genes regulating inflammation. And again, they picked out a whole bunch of sites across the genome that potentially are associated with inflammation and looked to see whether how the genes are associated with inflammation are methylated, and whether that methylation was associated with social position. These two papers reported that they were associated with social position, one arguing that it was early life social position that mattered for methylation of these genes, and the other one that it was later life that was important. It was sort of related to methylation of these genes. So even here you've got something quite complicated in that different parts of the life where it's most important to look. So there's a sort of an idea that a different way of doing this is to actually just look at across the entire genome and look to see if we can do it the other way around. So here we've got an example where people have looked at the entire genome and looked to see whether it's associated with methylation might be associated with age, which is the top panel there, or with smoking. So here what the group did, so Steve Horvath is sort of lead in looking at age and methylation. And what he did was one of the conventions in methylation and epigenetic work is that you do deposit your data. So but with very little additional information. So there are a lot of places you can go to get methylation data with the methylation data. And what he did was just use that information to see how methylation is changing with age. And he created a biomarker of age, which has been used actually recently quite extensively as an objective marker of age. And we have those data in Understanding Society. In the bottom panel there, people have looked to see if we can use methylation, how methylation is associated with smoking. So again, we just looked down the entire genome, and they used 450,000 sites across the entire genome and looked to see if it was associated with smoking. And then people have subsequently used the scores that have come from that to see if we can use biomarkers of smoking. We have those, we have the age biomarkers in understanding society, and we're making those available. We're going to deposit those in the archive soon. We look to see if Steve Horvath's, and there's a few of these biomarkers of aging. We look to see if Steve Horvath's biomarker of aging and another one called Hannum biomarker of aging were actually associated with age in our data set. And you can see in the graphs just at the top there that they are associated with age in our data set, but not in a parallel way. So our biomarkers of smoking were overestimating age in our youngest age groups and underestimating age in our oldest age groups, suggesting that our younger participants were biologically older than their chronological age would predict, and our oldest participants were biologically younger on average than their chronological age would predict. And so that's quite interesting, because these biomarkers of age were being taken by social epidemiologists to look to see how how they varied by whether you were biologically different to your chronological age was associated with, for example, the social position, and if you're using education to do that, then education means different things in different age groups, and if this difference in biological age and chronological age is age pattern, that's quite important. And so we published a paper suggesting that you need to be careful about how you're treating age in these analyses. And what we saw was that father social class was associated with being, so disadvantage in early life was associated with being biologically older than your chronological age. So you're aging more quickly biologically if you had experienced disadvantage in early life. We're also doing some work on how the biomarkers of smoking are functioning in understanding society. So this is some, at the bottom here, some unpublished work from a student, a SOCB student in our program, who's looking at how biomarkers of smoking are associated with smoking in self-reported smoking in our data set, and whether that what are the predictors of mismatch is. And you can see here that our data is suggesting that if you have lots of education, although fewer people report smoking, there is much more mismatch between the biomarker of smoking and self-report. So it looks like more people telling us that fewer people are telling us that they smoke than actually are if you're more educated. So we're sort of looking into that, sort of thinking about what that might mean. So what we're going to talk about next is this sort of omic approach to analysis, and what has been done in the social science setting with that. So there have been these sort of genome-wide association studies of things like educational attainment, risk behaviors, a number of, so smoking, alcohol intake, lots of behaviors, and people have used those and created these sort of scores, and we use these scores as and these scores have been used in approaches sort of approaches to analysis. So something called Mendelian randomization, which uses these scores as instruments in IV type analyses to see if we can kind of instrument the thing that we're interested in. And then there's also been newer work looking at epigenome-wide associations of these things, so educational attainment, and trying to understand sort of what biology is being uncovered by these epigenome wide approaches to social science variables. So the idea with this omic approach is that what you're going to do is you're going to look down the whole genome and kind of uncover new biology or support the candidate approach if it's there. And really that's what you're trying to do. What generally tends to happen is that you're kind of going down the genome and you find that some things are, say, for example, in this particular paper looking at educational attainment, they found that lots of sites were associated with educational attainment. Nothing is associated individually very much. And even when you add up everything that's associated with educational attainment, it isn't predictive of educational attainment, but the scores that you get from doing this work are kind of can be used. They're predictive enough to be able to be used as an instrument, and people have tried to sort of to use those properties of the scores that come out from this sort of work. I've been going on for too long. I will stop soon. This is my last slide. So what we're going to do next is talk about what the biology. So we'll go over again the biology that I kind of went through very quickly in my presentation. And then we'll talk about these sort of methodological approaches to the data and think about some examples. And finally, we'll present some what the datasets are and how you access them.